Assalamu alaikum students. Today we will discuss Golgi apparatus. Our topic from 11th class biology, chapter number 4, the cell. In this topic we will discuss about discovery of Golgi apparatus, its introduction, basic concept, formation and function. Golgi apparatus. It is named so on the name of its discoverer called Camillo Golgi. Golgi apparatus was discovered by Camillo Golgi in 1898. Before the start of that topic, we have to clear our concept regarding Golgi apparatus, Golgi complex. What is Golgi apparatus? Basically, Golgi apparatus is a combination of pouches. These pouches or flat sacs bounded by membrane called cisternae or Golgi body. These cisternae or Golgi body, when present in the form of a stack, then it is called Golgi apparatus. And when that stack have vesicles, then it is called Golgi complex. You can see here Golgi body or cisternae pouches, membrane bounded flattened sac or pouches. When present in the form of stack, it is called Golgi apparatus, and when that stack have vesicles, then it is called Golgi complex. This is the core concept of that topic. Let's start our topic Golgi apparatus. This apparatus, which was found virtually in all eukaryotic cells. Eukaryotic cell, as you know, all those cells which have well-defined nucleus are called eukaryotic cells. Consist of stack of flattened membrane-bound sacs called cisternae. These flattened membrane-bound sac, when present in the form of a stack, then it is called Golgi apparatus. These cisternae together with associated vesicles are called Golgi complex. When these cisternae are present in the form of a stack and they have vesicle, then it is called Golgi complex. So we can say that it is a complex system of interconnected tubules or sacs around the central stack. There is a main pouch and all other pouches are attached to it and it's interconnected. Each pouch is connected to next pouch. Formation. Basically, Golgi apparatus is formed from endoplasmic reticulum. From smooth endoplasmic reticulum, these membrane or cisternae are continuously formed by fusion of vesicles, which are probably derived by the budding of smooth endoplasmic reticulum. As you can see here, from smooth endoplasmic reticulum, these vesicles are budded off and they stack themselves to form the structure called Golgi apparatus. There are two phases of Golgi apparatus, forming phase and maturing phase. The outer convex surface is the forming phase, while the inner concave surface is the maturing phase. These phases also have another name, cis and trans. Phase that is present near to endoplasmic reticulum is cis phase, and the phase that is mature phase or exit phase or from where the vesicles are budded off after packing is called trans phase. So these phases have two names, cis phase, trans phase, and they are also called forming phase and maturing phase. Forming phase or cis phase is always present near to endoplasmic reticulum, and trans phase is mostly present toward the plasma membrane. The main difference between cis and trans phase of Golgi apparatus is that the vesicles that leave the endoplasmic reticulum fuse to the Golgi apparatus from its cis phase, whereas the vesicles that leave the Golgi apparatus exit from its trans phase. Vesicles, all the product that attach with Golgi apparatus, they attach from cis phase and then move from one cisternae to another cisternae and finally convert into finished product and then budded off in the form of vesicles, and it budded off from the later means from trans phase or from maturing phase. This is also a short question. From where the vesicles are budded off? The vesicles are budded off from the trans phase or maturing phase. Arrangement. The whole stack consists of a number of cisternae, thought to be moving from the outer to the inner phase. Outer phase, forming phase, inner phase, maturing phase. They are arranged themselves in this way that the new cisternae are attached on forming phase and mature cisternae or older cisternae are present toward the trans phase. Functions. Golgi complex is concerned with cell secretions. Now question arises, what are cell secretions? Secretions are products formed within the cell on ribosomes and mostly they are proteins and then pass to the outside through endoplasmic reticulum and Golgi apparatus. Cell produce secretions, mostly proteins, enzymes, and all these secretions then move outside the cell through endoplasmic reticulum or Golgi apparatus. That's why you can say its location, that endoplasmic reticulum, then Golgi apparatus. Ribosomes give its secretions to endoplasmic reticulum, and endoplasmic reticulum give its secretion to Golgi apparatus. Here you can see cis and trans phases. The secretions are received by cis phase and bodied off from trans phase. The next function is packing. Secretions are converted into finished product and are packed inside the membrane before export. Golgi apparatus convert that secretion into finished product and then pack into membrane and bodied off, as you can see here. For example, in mammal, the pancreas secrete granules containing enzyme that help in digestion. The Golgi complex has a role in the formation of these granules. Granules and enzyme pack in vesicles and then bodied off from Golgi apparatus. Golgi apparatus has a role in the formation of these granules. Next function is transport. The protein or enzymes which have to be transported out of the cell. Every kind of protein and enzyme which have to be transported out of the cell have to pass through Golgi apparatus. They can't transport it out of the cell without passage through Golgi apparatus. Golgi apparatus has important role in modification or in the formation of conjugated molecule. Mostly, it add carbohydrate with proteins or lipids and convert them into glycoprotein and glycolipids. The most important function of this apparatus is to modify the protein and lipid by adding carbohydrate and converting them into glycoprotein or glycolipids. Easy to memorize. If we have to memorize that topic, we have to divide that topic into two parts. The first part is related to its structure, the next part is related to its function. Whenever we have to memorize any topic, we have to keep in mind its diagram. Let's start. If we have to memorize that topic, we first of all we have to know that who discovered it or why it is called Golgi apparatus. Reason for naming. It is called Golgi apparatus on the name of its discoverer called Camillo Golgi. Then Golgi body, what are Golgi body, what is Golgi apparatus and Golgi complex. Formation, it is formed from smooth endoplasmic reticulum, then bodied off, and these cisternae stack themselves to form Golgi apparatus or Golgi complex. It has two phases, forming phase, maturing phase. Forming phase is also called cis, and maturing phase is also called trans. Forming phase is near to endoplasmic reticulum, and maturing phase is close to plasma membrane. Vesicles always bodied off from maturing phase. So these are the points related to structure. If we have diagram in our mind, we can easily attempt these through these points or easily memorize. Function, it has four function. It is related to cell secretions. What are cell secretions? Then packing, transport and modification. Whenever we have to memorize any topic, first of all we have to keep in our mind its diagram. Then divide that topic into two parts, structural portion and functional portion. With that, we have completed our topic. Thank you very much. Allah Hafiz.